osteosarcoma is a very deadly disease, and there has not been any progress made in the last 40 years. Previously there'd been a lot of work on chemotherapies, direct acting agents, and now these immunotherapies, these things that stimulate the immune system so that the immune system go kill the cancer, are taking increasing importance.
Hello, you're watching Proactive. I'm joined by Gerald Commissiong, Chief Business Officer at OS Therapies. Gerald, very good to speak with you. OS Therapies is described as the world leader in listeria-based cancer immunotherapies. Can you explain for new investors how your OST-HER2 therapy works? What differentiates your platform from other oncology treatments?
Sure. So if some of your folks remember, there was previously a company called Advaxis that spent about $300 to $400 million developing this listeria-based immunotherapy. We, through a series of transactions, have those rights. So for people who remember, it's basically you take listeria, it is attenuated so it doesn't replicate as much, and then we fuse it to an antibody so that it only infects cells that overexpress HER2. HER2 is a cancer antigen that sits on the cell surface of certain cancer cells, especially metastatic disease, and it helps with vascularization. And basically, once infected with the listeria, our vector releases additional cancer antigens, basically turning a 1+ into a 3+. Then that activates the immune system, and it recognizes the cancer cell, but also sees inside the cell that it's listeria, and it activates a different set of cellular immunity mechanisms other than just towards the cancer. So obviously, if the body was controlling the cancer, we wouldn't be talking about treatment. It's because of this lack of control and this incomplete immune response that cancer exists. And so what we try to do effectively is to allow for a more complete immune response in response to the infected cancer cells. That's the mechanism. And once it's infected, it's very similar to a company called IBRX from Patrick Soon-Shiong and his Anktiva drug. There he's trying to stimulate T cells and NK cells. And we also stimulate T cells and NK cells, as well as dendritic cells and some other immune cells to go and attack the cancer. So that's the mechanism of action, is basically activating cellular immunity against the cancer by tricking the body into thinking that, cancer is listeria and listeria is cancer.
So, you've announced quite strong progress over the past few weeks, Gerald. You've just announced a successful Type C meeting with the FDA. Also recently, a pre-marketing authorization application meeting with the UK's MHRA. What were the key takeaways from these discussions, and how do they shape the regulatory path for OST-HER2 in osteosarcoma?
Basically, the tenor of those conversations, as well as, we had a good meeting with the European CHMP not too long ago. They're all around trying to find a pathway to get this drug into patients' hands sooner, because osteosarcoma is a very deadly disease, and there has not been any progress made in the last 40 years. Previously there'd been a lot of work on chemotherapies, direct acting agents, and now these immunotherapies, these things that stimulate the immune system so that the immune system go kill the cancer, are taking increasing importance. So for us, in the US, what we really got to was the meat of the situation on how the FDA could use the data generated in our single-arm study to be able to get to an approval. There have been a lot of questions about whether or not historical control could be used as a suitable comparator. They basically opened the door to saying, well, potentially it could be if there's sufficient effect size and if there is evidence that the treatment effect seen is directly attributable to the drug. And that's where the biomarkers come in. So you can see within the patient, hey, they don't have, some threshold of T cells or NK cells. And then after a number of treatments, those number of T cells and NK cells have gone up. And that has led to a good clinical outcome. So that's really has been the tenor of the conversation with all the agencies is, the data looks pretty interesting on the surface, but there's no concurrent comparator. So how are we able to determine that the treatment outcome is, as a result of your drug? And so, we've been working with our KOLs, key opinion leaders, and with the regulatory advisors and other to come up with a strategy, and we're making progress in that direction.
What's the next steps to take your platform forward, Gerald?
So I like the word platform, so thank you for using that. So it's important to note that OST-HER2 is one of several constructs. So we have a OST-AXLA, which is really targeting HPV-driven cancers. We also have OST-503, which has a number of different cancer antigens, and OST-504, which is going after prostate cancer antigens. So the next step for us is to complete the biomarker analysis, do the correlation work, submit that to FDA, and then submit for approval, and then have meetings as part of that approval process to, one, confirm that they agree with our assessment once we have it. If the biomarkers show correlation with clinical activity, then we have to get them to agree with that, and we can't do that yet until we have that data, but we expect that data out within a month's time, probably in and around JP Morgan. We will then also be we're talking now about accelerated approval, not full approval. So in order to get accelerated approval, you have to have already designed and initiated your confirmatory study. So we don't need to have dosed hundreds of patients or anything like that. We just need to open a site and start the enrollment process. So not very expensive, but it means we have to have thought through all the issues that would ultimately be required for that confirmatory study. And so that's what the next big thing. The analysis part, we have our biomarker stats people that are working in that direction, and then separately, once we have that data, we already have people designing or thinking through the design. We have a synopsis, and obviously we discussed it with the regulators. And as part of that design, at the end, you have to take the stats that we get, in order to create an effect size to determine how large the confirmatory is going to be. So, those things go hand in hand, but they're happening in parallel.
Gerald, I hope you'll keep us posted on progress on all of those fronts. Thank you very much for speaking with us today.
Thank you.
That's Gerald Commissiong, Chief Business Officer at OS Therapies.